is Kathryn Adkins, and today I'm going to be presenting my bacteriophage poster. I did Dr. Adair's Bears in the Sea program, and in this class is basically, instead of doing a normal Biology 1 and Biology 2 lab course, I was able to research bacteriophages. So let's dive on in. Bacteriophages are basically viruses that infect bacteria cells. Our lab specifically looked at Arthropacter. Arthropacter is a soil bacteria. There are many different type of phages out there. They're all very specific. That's the different one. And what's awesome about bacteriophages, or what we were doing in our lab, what our goal was to isolate and characterize different arthropacteriophages found in specifically Arthropacter, and to understand their diversity and function. And we also wanted to compare phages based on bioinformatics. So my lab first collected soil samples, and one of our soil samples collected near the Brazos River contained our phage Napoleon B. After Napoleon B went through different phage DNA extraction procedures, and we were able to take our DNA and we sent it off for Illumina sequencing. So that's the one that does all the bridging, folding over. And so once our DNA was isolated and sent off for sequencing, we were able to use different computer programs such as DNA Master and all these different databases to determine its function. So let's look at our wet lab results. So we did some metadata where we basically analyzed the soil. We didn't really come across any distinctions between, like, we didn't see that all our phages had the same type of soil or similar soil. We didn't really discover anything like that. Napoleon B, what's really interesting about Napoleon B is most phages only produce one plaque size because, as I said before, phages are super specific and to their hosts and only do like one distinct plaque. But Napoleon B did two different plaque sizes. He did these small ones, right? These small plaque sizes and these bigger plaque sizes. Plaques are little clearings showing where basically bacteria burst from all the phages being reproduced inside it. So those are basically dead bacteria cells. We also did a TEM image of Napoleon B. So let me, so this right here is, okay. So this is the phage's capsid, which is basically, or also known commonly as the phage's head. All the phage's DNA is inside of the capsid. This phage is a schizoviverte, meaning that it has a capsid and this long phage tail, as I'll just color over it. And this is the tail that's used to penetrate the bacteria cell. So that's what it looks like underneath a microscope. and this is really tiny. So this bar line shows, this is 100 nanometers. So phages are super small. We did gel electrophoresis to determine what type of family or which cluster, which is basically a family or grouping, this Napoleon B was a part of, and we discovered it was a part of cluster AM. Let's look at some of our in silico results, or computer analyzing bioinformatics stuff. This right here, this rainbow thing, this is a famirator, and basically this long line in the middle is Napoleon's B's genome, and we are comparing it to 2 other phages in the same cluster. Each one of these rainbow squares represents a different gene, and the purple shows high similarity. So this gene, so it has the same syntax, same gene, same function, etc. So we were able to determine that we were able to determine based on similarity to other AM phages, and also looking at this program called GeneMark, we were able to see that there's like, oh, there's actually a promoter here for this gene, and we were able to basically correct what the computer originally predicted as genes. So we inserted this gene because there was a gene here, there was and we saw the high similarity. We also inserted a gene right here and one right here. We were also able to determine genes that were incorrectly assumed by the computer program. For example, gene 30 and gene 46, both these genes, since they're on this other side, they're reverse genes. All these other genes on top are forward genes. One common rule, if you ever look into bioinformatics, is if you have genes like all together, like a bunch of genes like this, and they're all going one direction, it's very unlikely that you'll have one random gene going the opposite direction, because with all the promoters and stuff, it's just unpredictable. And we also were able to eliminate it based off the other 2 phages we compared it to. They didn't have any reverse genes in this section. So based off our genes, we were able to predict different functions. We were only able to predict 1/4 of the functions. There was still a lot of functions we weren't able to determine, but we were able to determine all these functions thanks to all these different databases. So NBCI, most people are familiar with this. HHPrep is basically, it's like BLAST, but it's for proteins. So it blasts proteins that are already folded and compares it to each other. PhagesDB is the database that we submit all the phage data is submitted to, and CDD is a part of NCBI. So right here, this is probably the most interesting thing we found in our research. So this goes back to, at the beginning, I was talking about how phages are specific and they have all this diversity. Well, basically, we were able to, when we were seeing all the different phage functions and comparing it to other genes, we saw that, of course, most of the genes that Napoleon B had, other phages had in its cluster. But we also see this red, all this red. These are phages these represent phages outside Napoleon's bees cluster that had the same gene in the same, relatively same location with the same function. So what that suggests is that there might be superfamilies or superclusters, and maybe phage categorization should be reconsidered to have just overall big superfamily clusters with all, and then you can have a specific cluster based off, a subcluster based off that. Okay, my computer's acting weird. Okay, there we go. So in conclusion, you might be wondering, okay, Kathryn, phages are really cool because, you know, viruses and everything, but why do we care? I want to address this. Bacteria, most viruses are commonly perceived negatively, but however, the phages, bacteria, the phages that the database we submitted our phage data to was eventually used to make this phage cocktail, which is basically a treatment where they mix a bunch of different phages together. And this treatment was used to treat a girl who had cystic fibrosis, stage 4, who went to the hospital, had surgery, and unfortunately, having surgery is always like a last result because she got infections. She got bacteria infections. So this phage cocktail that they were able to use based off the data we collected was able to save this girl's life. So I think that makes phages super awesome and super cool. Does anyone have any questions for me?
Yeah, where did you collect the samples again?
My class found 8 phages my year, and they were just all over Baylor. Napoleon B, the one that we specifically looked into its genome, was found right outside the football stadium by the Brazos River. I found my bacteriophage outside the marina. I mean, when you're looking for bacteriophages, you have to think, where would you find the hosts? Arthobacter is a soil bacteria. So after collecting 5 soil samples, I asked my TA, hey, where should I look now? And he told me to look somewhere dirty. Bacteria love to be in dirty places. So I went to the marina outside this, like, they have this like pipe. I know it's not a sewage pipe, but it smells like one. And that's where I found my phage. Yeah.
Okay, before you guys go off to the next.
Any other questions?
Yes, while Kathryn asks or answers any of your other questions, let me send this in the chat so that you guys can sign out for attending this breakout session. But feel free, if you do have any other questions, Kathryn can leave her email in the chat, and feel free to email her anything, or you can stay for this presentation once more and then ask it again in the ending session.
Oh, and also if you're needing a journal club credit, I will be hosting a phage journal club in the next 2 weeks. Yeah. I love to talk more about phages.